So, nucleic acids. Nucleic acid is the most important molecule out of the main biomolecules in living matter in genetical aspect. So, this is a special thing new to y'all up to now. We didn't discuss about the genetical aspect. We saw that carbohydrates, proteins and lipids are usually structural components. They provide energy, but none of them had this genetical aspect. When we say genetical aspect, we talk about genes and characters being transferred from one generation to the other. Now, if you think of yourself, you look at the features, you will resemble maybe your parents or your grandparents. That is, you have characteristics from the generation before you. So, this transfer of characteristic from one generation to another is known as genetics. And this is the genetical aspect of nucleic acid, because nucleic acids are the biomolecules that are going to store information related to characteristics and transfer it from one generation to another generation. So, if we look at the structure of a nucleic acid, it is a linear polymer. Linear means it's like a line, like a thread, no branches, no cross links. So, it is a line and it is a polymer. Polymer means many, a large molecule that is made up of many small molecules. So, nucleic acid is such a polymer. It is a linear polymer, and this is made up of a large number of nucleotides. So, the basic structural component of nucleic acids are called nucleotides. Many nucleotides combine together and form a linear strand. So, therefore, these are linear polymers. And in nucleic acids, there is carbon, hydrogen, oxygen, nitrogen and phosphorus. So, there are 5 elements: carbon, hydrogen, oxygen, nitrogen and phosphorus. So, these 5 elements combine in different ways and they form 3 different components. Those 3 components combine together and form the nucleotides. Then these nucleotides form the large polymer structure which is called the nucleic acid. And if you remember, students, when I discussed the significance of carbohydrates, I said that it is a constituent of nucleic acid. There I mentioned pentose sugar. Pentose means 5-carbon sugar. So, based on the type of pentose sugar present, there are 2 types of nucleotides. And based on these 2 types of nucleotides, there are 2 types of nucleic acids. And now we will look at the structure of a nucleotide. So, here as you can see, as I said, carbon, hydrogen, nitrogen, oxygen and phosphorus combine and form 3 components. One is called the nitrogenous base. So, from the name itself, you know that there is nitrogen present in this base. And there are different types of nitrogenous bases that are present in the nucleic acid. And there is a pentose sugar. Pent means 5. Because there are 5 carbons, it is called a pentose sugar. And the third group, which is a phosphate group, has a PO4 3- group. That is what we call as phosphate group. So, a nitrogenous base, a pentose sugar and a phosphate group combine and form the nucleotide molecule. And these are usually the standard shapes given for these molecules. This is a pentagon to denote that there are 5 carbons and the nitrogen base so that it can combine with another nucleotide chain and also the phosphate group. Now we will see, based on the different types of pentose sugars present, there are 2 pentose sugars that are present in nucleic acids. Therefore, there are 2 types of nucleic acids. Here you can see one is called deoxyribonucleic acid. If we take the first letter of these 3 words, DNA, deoxyribonucleic acid. So, DNA is one of the nucleic acid. The second one is called ribonucleic acid. Taking the first letters of the 3 words, we get RNA. Now, this is based on the type of pentose sugar that is present. So, in a DNA molecule, there is deoxyribose sugar. This is the pentose sugar group. And in an RNA molecule, there is ribose sugar. These are the 2 pentose sugars. If it is deoxyribose sugar, we call it a DNA molecule. If it is a ribose sugar, it is known as an RNA molecule. Now we will look at the structure and the significance of these 2 molecules. So the DNA is the structural unit of DNA is the ribonucleotide. So as I said, the combination of a phosphate group, a pentose sugar and a nitrogenous base is called a nucleotide. Many nucleotides combine to form the nucleic acid. So when deoxyribonucleotides combine, they form the DNA molecule. And the importance of this DNA molecule is to transfer genetic characteristics from generation to generation. As I explained before, we and all the other organisms, man and animals, plants, all the organisms get their characteristics from the previous generation. And the information regarding characteristics are stored in DNA. The second type is the RNA molecule. Except DNA, the other nucleic acid present in organisms is RNA. So, because there is ribose sugar, the nucleotide becomes ribonucleotide, and when the ribonucleotides combine, the RNA ribonucleic acid will be formed. And here, the function of an RNA molecule is protein synthesis. We saw DNA transfers genetic characteristics from generation to generation, and RNA is involved in protein synthesis. Also, you can see here, this is the structure of a DNA molecule. As we discussed before, if you can remember, students, nucleic acids are linear polymers. Linear means it's like a line. So here you can see this red color strip is one strand of nucleotide chain, and the blue color one, the ribbon-like structure, is another strand of nucleotide chain. They are both polynucleotides and they combine together and form a ribbon-like structure, which is the nucleic acid, deoxyribose nucleic acid. and this deoxyribose nucleic acid folds like a twine and forms a structure like this, which is called a double helix structure. This is the structure of a DNA molecule, and these, which are denoted by different colors, are 4 different nitrogenous bases. You don't have to go into detail about the types of nitrogenous bases, but you just need to remember that there are 4 types denoted as A for adenine, T for thymine, C for cytosine, and G for guanine. So these are the 4 different types of nitrogenous bases present in DNA molecule, and that is the double helix structure. If I write it here, it is the double helix structure. Of DNA. We will be discussing this structure in detail in later units. Now we will look at the significance of these 2 nucleic acids. Significance of nucleic acids important in storage of genetic information of organisms. So as we saw before, DNA is responsible for storing and transferring genetic information, and also it is important in transferring genetic information from one generation to another generation. So DNA is the main molecule for that process. As you can see, it is important in protein synthesis. That is the RNA molecule. Only the RNA molecule involves in protein synthesis. If you look at the next one, important in controlling all cellular activities in a cell. Now, if we look at the cell structure, which we will be discussing in detail later, there is a nucleus. Inside the nucleus, there is DNA, and this DNA controls the activity of the cell. So, therefore, the information to control cellular activities is present in DNA. The next one is RNA is important in storing genetic information in some viruses. This is something you have to remember. Normally, DNA stores and transfers genetic information, but in some viruses only RNA acts to store and transfer genetic information. RNA stores genetic information in some viruses only. And the last one is variations that occur in DNA due to mutations are important in evolution. Now, there are many terms that you need to understand here. The first one is variation. Varying means change. Now, we saw in the previous slide, the DNA molecule has a certain order of nitrogenous base sequence. So, that is responsible for certain characteristics. So, let's say father has curly hair and mother has straight hair. So, the child will either have curly hair or straight hair. That is how we get information or characteristics from the previous generation. But if you have a new type of hair, that is a change. That is what we call as variation. A change to the existing character is known as variation. Now, this variation occurs due to mutation. Mutation means there is a change that has happened to the DNA molecule that stores genetic information. So, this change is known as mutation. So, that mutation will lead to the formation of new characteristics, which is called variation. Now, these two are important in evolution. When we say evolution, it is the process that has been taking place from the time the earth evolved. You all know on earth it was first the living unicellular, one cell organisms that were formed. From that one cell organism, now if you look around you, there are so many different types of living organisms. Many types of animals, plants, unicellular organisms. So, it is a very wide variety of organisms. Now, all these happen due to changes. That process of development and change that took place naturally is called the process of evolution. So, all that was possible because of this DNA molecule. So, as you can see, the variations that occur in DNA molecule due to mutations in the base pair sequence are important in evolution and existence of diversity in the world. Diversity means difference in organisms. So, from this, you would have got a very good understanding as to how nucleic acids are important in the genetical aspect. So far we have discussed the 4 types of biomolecules, the carbohydrates, proteins, lipids and nucleic acid. I am sure you would have got a good understanding of what these molecules are and how significant they are to living organisms. Now, we will be looking at 3 different tests. The test for presence of water, test for the presence of carbon and the test for the presence of nitrogen. The first test identification of water as a constituent in food. And these are the materials required. I will explain the process along with the materials that are shown here. We will need an organic material, that is a plant part or an eggshell which contains water. We will be using one of these substances for our test. So, here I can say a plant leaf or eggshells. This is the material that we are going to test the presence of water as a constituent particle. So, what we have to do is we have to take these parts, say leaves of plants or the eggshell, but it has to be in the dried form. Dried form in the sense we don't have to dry it in the sun, but we shouldn't wash it and take it. Because if you wash the leaf will have water on its surface. So, we wouldn't know whether it is the water on the surface or the water that is present in the leaf. So, therefore, the outside, the external side of the substance that we are going to use should be dry, should not have water in it. So, that is the substance that's going to be tested. And the chemicals that we are going to use today are 2 chemicals. One is copper sulfate. As you know, the anhydrous copper sulfate is white in color. Here the picture shows the hydrated copper sulfate. Usually this is the copper sulfate that is available in the lab. So what we can do is we can take the hydrated copper sulfate, heat it using the Bunsen burner, then we will have the anhydrous copper sulfate. So this is one of the chemicals, copper sulfate. We will heat and take the white color anhydrous copper sulfate. The chemical formula of copper sulfate is CuSO4. So I have written both so that you will remember the chemical formula as well. So we will be needing the white color anhydrous copper sulfate for the test. The procedure is going to be, we will take the part of the plant or the eggshells, we will put it into a mortar and a pestle. By now, students, you are familiar with the mortar and the pestle. We use this equipment to grind substances. So, we take the plant leaves, take it into the mortar and the pestle, and using the pestle, we grind it into tiny parts, small particles. It doesn't have to be really powdery, but small parts. If you can easily tear the leaf into pieces also, that should be okay. You can make it into very tiny pieces so that it will be easy to heat the substance. Then we will be using this particular equipment. This is called a crucible. The crucible is a small porcelain container with a lid. We use this to heat samples for a very long time or to very high temperatures, because this crucible can withstand high temperature over a long period of time. So, we will transfer the ground plant parts or the eggshells into the crucible. Then we need to use the Bunsen burner and the tripod with the wire mesh for heating. So, we will need the Bunsen burner, tripod and wire mesh. So, we set the tripod to the Bunsen burner, light the burner, and then we place the crucible on the wire mesh with the lid cover. We leave it to become hot for some time. And after some time, once you feel that this has become hot enough, what we do is we open this lid, and this is a piece of glass. We hold that piece of glass on top of the crucible. So, the water that is there in the food sample will evaporate due to heat, and when we hold the glass on top of it, it will condense on the glass. So, for that we will be needing a glass sheet. So, then what do? We take the glass sheet that contains water droplets, and onto that we put the anhydrous copper sulfate which we have already prepared. Then you will get the observation. The white color of the copper sulfate will turn to blue in color. So, as we discussed, we can identify whether the vapor that is produced is water or not using anhydrous copper sulfate. In addition to this, we can also use another chemical substance, that is anhydrous cobalt chloride. Anhydrous cobalt chloride is blue in color. When it comes in contact with water and becomes hydrated cobalt chloride, it becomes pink in color. So we can also use anhydrous cobalt chloride to identify that the liquid vapor that condensed on the glass sheet is water. So I will include that reagent here. We can use the blue color anhydrous cobalt chloride. To test and confirm that the vapor that came out from the food sample and condensed on the glass sheet is water. So students, now that you have understood what we have to do here, I will write down the steps of the method of this activity. So the method is going to be the first step, crush the food sample or plant part using the mortar and pestle. We have to make sure that we use a dry substance. So I will write that in brackets. Use dry materials so that there is no water on the surface. Then what do? We need to prepare anhydrous copper sulfate. Prepare anhydrous copper sulfate by heating hydrated copper sulfate. So we have the white color anhydrous copper sulfate ready. We have the plant part ready. Now we will heat it using the crucible. So transfer the crushed plant parts or food sample into the crucible. Then the next step, we will heat it with the cover. Cover the crucible . Cover the crucible with the lid and heat using the Bunsen burner. So once we heat the sample using the crucible on a Bunsen burner, when we open the lid, you will see that there is a vapor coming out. So what we do is we hold the piece of glass on top of the vapor so that it will condense on the glass. The next step, remove the lid and hold the glass so that the vapor condenses on it. So now we have got the drops of liquid condensed on the glass piece. The final step is to test and confirm that the vapor that condensed is water. So to do that, we have to test it with anhydrous copper sulfate. So the last step, test the liquid drops with anhydrous copper sulfate. So test the liquid drops with anhydrous copper sulfate. In addition to that, I told you all that we can use anhydrous cobalt chloride. So we can also test the liquid drops with anhydrous cobalt chloride. So I will add that also here, and anhydrous cobalt chloride. So here we will be doing the activity and testing the liquid droplets with both chemical substances, anhydrous copper sulfate and anhydrous cobalt chloride. So students, now that you have understood the steps of this activity clearly, let us do the activity in the lab. Okay students, now we are going to do an activity to test the presence of water in food samples. For this, you can use any type of leafy vegetable or fish, meat, or even any type of leaf, because all living organisms contain water in their body. Today, I am going to use some leaves from a garden plant. So, these are the leaves, and we should make sure that the sample, if it is a leaf, that we don't wash it prior to the activity. If we wash it, we wouldn't know whether the vapor that is coming out is from the water that is there on the surface or from the sample. So, we will have to make sure that we use a dry sample. And then we will have to heat this for a long time. So, I am going to use a crucible with a lid, a watch glass to make the vapor to condense on it, and we have anhydrous copper sulfate which has to be white in color. Also, we have the pink color cobalt chloride solution, but we will have to dip a piece of the filter paper in cobalt chloride, and then I will heat it so that it becomes anhydrous cobalt chloride, which is blue in color. So, the reagents here are anhydrous copper sulfate and anhydrous cobalt chloride that will be made from this. Now, to start the activity, I will take the crucible. Then, into the crucible, I will put few pieces of the leaf. If we break it into pieces, it will become hot quickly. Then what we do is we place it on the tripod on top of the wire mesh and I will close it with the lid. After it gets heated up for some time, we will remove the lid and cover it with the watch glass so that the vapor that is coming out will condense on this watch glass. It has been heating for some time. I will remove the lid of the crucible using a pair of tongs and place this watch glass over it so that you can see the vapor coming out and condensing on the watch glass. We will collect that vapor. Now you can observe, students, that there is a liquid that is vaporizing coming out of the sample and condensing on the watch glass. I will remove the watch glass now. You can clearly observe the droplets of liquid, and to that I will put copper sulfate. You can see the color changing. The light color of the copper sulfate, that is anhydrous, is turning to dark blue color. At the same time, I will dip a small piece of paper. When I hold it to the flame, you can see the pink colored cobalt chloride. You can compare and see becomes blue color. If I touch it on the drops of liquid, again you can see it changes color. So here you all can see, students, the copper sulfate has become blue in color. This blue part is the anhydrous cobalt chloride. Wherever there was water, it has become pink in color. So these two observations confirm that the vapor that came out and condensed on the watch glass is water. So with this observation, we can conclude that there was water present in the leaf sample. That means there is water in food samples as well as living organisms. Okay, students. So you observed the color changes clearly when we did the activity in the lab. There was a liquid that condensed on the watch glass, and when we put the white colored anhydrous copper sulfate, it became blue in color. And when we touch the liquid droplets with a strip of paper that had blue color due to anhydrous cobalt chloride, it turned pink in color. So those two observations, the white color anhydrous copper sulfate becoming blue in color, the blue color anhydrous cobalt chloride becoming pink in color, are confirmations that the liquid is water. So we will first write down the observation of the activity. White colored anhydrous copper sulfate turns blue. At the same time, the blue color anhydrous cobalt chloride turns pink. This confirms that the liquid drops that condensed on the watch glass is water. This confirms that the liquid drops is water. Therefore, we can conclude that the food sample or plant part contains water. So students, we carried out this activity to test the presence of water in food samples. So you saw the observations clearly and you have understood the procedure and the conclusion of this activity. Now let us do the second activity to test for the presence of carbon in biomolecules. So the test, identification of presence of carbon in biomolecules. So as you all know, the plant parts, food items, they are all made up of biomolecules. So if we test for the presence of carbon, say in a food sample or a plant part, we can confirm that there is carbon in biomolecules. So the sample that we can use can be a vegetable, a leafy vegetable, or a cereal like this, or else we can use any plant leaf or a small plant part. So here for the sample I will write it as a plant part or food sample. So the first step of the activity, what we will be doing is, we will take a food sample. If it is kadala, you might have to soak it in water so you can grind it easily. Or if it is a green vegetable or just a leaf of a plant, then it would be easier to grind. So as usual, we will be using a mortar and pestle to grind the food particle. So we take the food sample or the leaves and break it into small pieces, put it into the mortar and pestle. We can add a little bit of water and grind it nicely, make it into a pulp. Pulp means a semi-solid substance that is ground nicely. It will be like a paste. So then we use the tripod with the wire mesh and the Bunsen burner to heat this sample. So for that, what is the glassware we have to use? Yes, we have to use the crucible. The crucible is the laboratory glassware that can be used for prolonged heating or heating at high temperature for a long time. So we will use the crucible. We transfer the pulp, the food ground pulp, into the crucible, place it on the wire mesh of the tripod, which is set with the burning Bunsen burner. So we will be needing the tripod, wire mesh and the Bunsen burner. So we will use the burner to heat the sample for a long time. And on and off, we will have to open the lid of the crucible so that the water vapor we tested in the previous activity, the water vapor will go out. We just open it and close the lid, let it to get hot for some time, then again open the lid, close it, leave it until you see the sample becoming black in color. So after a long heating, you will see the pulp, the food sample that was taken will turn to a black color residue. When we say residue, it is the solid substance that remains after a long heating. So then what we do is we need to use a pair of tongues to remove the crucible from the burner because it will be very hot. So then you take it off the burner, leave it aside for some time so that the sample also cools down a little bit so that you will be able to touch it. Then you can use a spoon or a spatula to take out the residue, and we have a sheet of white paper here. We need a sheet of white paper. Now the residue is cool, you can touch it. You can take the black color pieces of residue and you can just rub it over the white paper and you can get the required observation. So students, I hope you understood the steps related to the activity to test the identification of presence of carbon in biomolecules. I will again summarize the steps to you under procedure. The method. So what do we have to do first? We have to make a pulp from the food sample or the plant part using the mortar and pestle. We put the food sample into the mortar and pestle, add a little amount of water, crush it well, grind it well and make it into a pulp. We'll write that down first. Make a pulp of the food sample by grinding it with water. We can use it as a pulp, or else if it is a plant leaf, we can just tear it into smaller pieces and you can heat that pieces. So we can either use the pulp of the food sample, or if we are using leaves of plants from the garden, we can just tear it into small pieces and heat the leaf. Both are possible. I will include that also. Can use small pieces of leaves. That is also possible instead of using a pulp. Then what do? We take the plant material into a crucible. Why a crucible? Because it can withstand very high temperature. Transfer the sample into a crucible and heat. While heating, what do? On and off, we remove the lid of the crucible so that the water vapor escapes from the food sample and it becomes dry. And so that we can get the carbon as the remaining substance. So then you have to remove the lid of the crucible to allow water vapor to escape. We have to keep on heating until we get a black color residue. Heat until a black color residue is obtained. Then what do? We allow the black color residue to cool down. After that, we rub the black color residue on a sheet of white paper. So, rub the black color residue on a sheet of white paper. Then you will get the required observation. So students, now you have understood the steps of this activity clearly. We will carry out this activity in the lab. Okay students, now we will do the activity to test for the presence of carbon in food samples. For this again, you can use any solid food sample, leafy vegetables, piece of fish, meat, anything can be used. Today I am going to use these leaves. I will take a few of these leaves, and you all know for high heating we will be using a crucible with a lid. I will remove the lid of the crucible and break these leaves into pieces and put it into the crucible. Place it on the wire mesh of the Bunsen burner and place the lid. While it is getting heated up, we will have to remove the lid on and off so that the water that is vaporizing from the leaves will be removed. I will remove the lid on and off. You can see the water vapor coming out. Again, close the lid. Leave it for some time. Again, I have to remove the lid and allow the water vapor to escape from the crucible. Like this, we have to keep on heating it until there is a change in the substance. Again, if I remove the lid, you can see that the leaves are turning in color. They are almost becoming brown in color, and when we keep on heating it, they will become more and more dry, and finally we will end up with our required end product. I will close it again and let it to heat. Now again I will remove the lid. You all can see students that the sample has become black in color. We will leave it for few more seconds. Yes, you can observe nicely there is a black color residue that has been formed inside the crucible. Now that all the water has become vapor and has been removed from the sample, there is only a black color residue remaining. Then I will have to remove the crucible from the burner. You will have to leave it for some time because it will be very hot, and after that, once it cools down, we can take out a small piece of the black color residue. And then I will try to draw lines on this white filter paper. I think by now the sample must have cooled down a bit. I will take out the most blackish color parts, place it on the paper. You have to make sure it is not hot. Then I will draw some lines with this. Here you can see clearly that there are black colored lines being drawn on the filter paper. By now you would know what it is. You have seen the observation. Black color is carbon. Carbon graphite, what we normally get from burning firewood, is also black in color. When you burn firewood, we end up with coal or charcoal. So this charcoal, if you draw it on a piece of paper, you will get similar black colored lines. So from this observation, we can confirm that the plant leaves have been converted to charcoal. Charcoal means they are in the form of carbon. So that confirms there was carbon present in these plant leaves. Okay students, now you had a good view of the activity that we did in the lab to test the presence of carbon in food samples or in biomolecules. Can you remember the observation clearly? You saw it properly? Yes. When you draw, you spread the residue on the white sheet of paper. You see black color lines being drawn on the paper. So these black color lines are caused by coal. Coal means carbon. So if there is carbon, that means it came from the food sample that we tested. So that will confirm the presence of carbon in the food sample that we used. So to write down the observation and conclusion, observation will be black color lines will be drawn on the white paper. Black color lines will be drawn on the white paper. Because the black lines are due to carbon, we can confirm the presence of carbon in the food sample we used. So the conclusion will be the sample tested contains carbon. So students, this is the activity to test the presence of carbon, and you have understood the steps and the observations clearly. Now we will go on to the next activity, the activity to test the presence of nitrogen in biomolecules. So test identification of presence of nitrogen in biomolecules. For this activity, we will need all these materials, and as the biomolecule, we can use any one of the food that is rich in nitrogen. Because you can remember students, the 4 types of biomolecules: carbohydrates, proteins, lipids and nucleic acids. And out of those, proteins and nucleic acids contain nitrogen. And usually the proteins, because of the amino acid structure, have high amount of nitrogen in it. So it is easy to obtain a food that is rich in protein. For example, a piece of fish or the egg white. So for this activity, we can use a piece of fish or egg white. So this is a food rich in proteins, therefore there will be nitrogen. Then the activity is done by taking, if we are using egg white, we directly take the egg white into a test tube. But if we use fish, then we will have to grind the fish using the mortar and pestle. So for that, we will need mortar and pestle. Egg white can be directly taken into the test tube. If we use a solid food, then we have to grind it and take the extract into the test tube. Here we will need more than one test tubes, because if you can remember, we did this activity before, identification of proteins in food. So then for that we will be needing another 2 reagents. So we will need more than one test tubes. So here these are the test tubes that we need for the activity. The reagents that are needed are copper sulfate solution, and sodium hydroxide solution. And to make the food pulp, we needed water. And also to dissolve the copper sulfate and sodium hydroxide. So as you know, in the lab, we have the normal hydrated copper sulfate that is blue in color. We take a small amount of copper sulfate into a beaker, add water, and we can dissolve it into a solution. The same way, we can either have the sodium hydroxide solution in the lab, then we can use that. Otherwise, you can see the sodium hydroxide pellets. Those are white color pellets. We take a few pellets into a beaker and dissolve it with water. So you will have the sodium hydroxide solution ready. Then what do? We already have a sample of the protein containing food, either egg white or extract from the fish. We will have to measure that volume, and equally we will have to take sodium hydroxide. So into 2 test tubes, one will have the food substance and the other one will have the sodium hydroxide solution. The 2 volumes have to be equal. Then we transfer the sodium hydroxide solution into the food sample. And afterwards, we can add a little amount of copper sulfate solution. The copper sulfate solution is blue in color, but as you add it to the reaction mixture, you will see a color change. So that will be the observation. You would have understood the steps in carrying out this activity. We will summarize it and write it down as the method. So, under method, if it was egg white, you can use it directly. If it is a solid food, you will have to crush it and take the extract. So, the first step is to crush and get the extract of then you will get the required observation. Now that you understood the steps clearly, we will do this activity in the lab. Okay students, now we are going to do the activity to test the presence of nitrogen in food samples or the presence of nitrogen as a constituent in biomolecules. We have already done this test with proteins. You know the name of this activity. What is it? Yes, it is Biuret test. So, I will be needing a sample that has protein so that we can test for the presence of nitrogen because nitrogen is a constituent of protein. The reagents, the two reagents, sodium hydroxide solution and the blue color copper sulfate solution. I have already taken the egg white into a small beaker. Now I will transfer the egg white into a test tube. Okay. Now I will take another test tube and into it the same volume of sodium hydroxide or slightly excess volume of sodium hydroxide has to be taken. Here you can see students, it's almost the same volume, if at all a slightly excess amount of sodium hydroxide. Now I will transfer the sodium hydroxide into the egg white. To this mixture, we will add a few drops of copper sulfate. Now you can see the observation clearly. Can you all identify the color change, the observation? Yes, the appearance of purple color. This confirms the presence of protein in the sample, and you know that protein contains nitrogen as a constituent element. So therefore, we can conclude that this sample contains nitrogen in it. Okay students, now you saw the observation of the activity that we just did to test the presence of nitrogen in biomolecules. What was the observation? Yes, as you add the blue color copper sulfate solution, the reaction mixture becomes purple in color, or the formation of purple color indicates the presence of nitrogen in a food sample. How? Normally, this test is called the Biuret test that we do to identify the presence of protein. So, proteins contain nitrogen as their constituent element. Therefore, the presence of purple color or the formation of purple color indicates the presence of protein, and that confirms the presence of nitrogen in the food sample. So, the observation is the appearance of purple color. Purple color. The conclusion is the purple color formation in the Biuret test confirms the presence of protein, therefore the presence of nitrogen. So, I will write the conclusion as the formation, the appearance purple color in Biuret test confirms the presence of protein. Therefore, the food sample contains nitrogen. So that is the activity to test for the presence of nitrogen in biomolecules. We do this test with copper sulfate and sodium hydroxide. This is called the biuret test, as we did for proteins, and the formation of purple color indicates the presence of protein, and that in turn indicates that there is nitrogen in the food sample. So students, now you got a good understanding about the 4 different types of biomolecules, their presence and the significance to living organisms, and also the different types of activities done to test the presence of these biomolecules, as well as these 3 components: water, carbon and nitrogen. So that is the section related to the organic compounds present in living organisms. Now we will get on to the next section where we talk about inorganic compounds like water, minerals and also the organic molecules, vitamins. So first we will see how significant water is to living organisms. Water, the highest proportion of the body mass of living organisms, is composed of water, which is an inorganic compound. So here we have to note highest proportion of the body mass, right? So if you take any living organism, whether it's a unicellular organism, a plant or an animal, you look at the structure of its body and you will see that the main component, the higher percentage of the body will contain water. So therefore, the body mass, that is the mass of the living organism, what we measure in kilograms, the mass, the main proportion of the body mass will be water. That is why when we get the mass of living organisms, we call it as the wet mass, because there is lot of water in the living body of organisms. And also another point that you need to note, it is an inorganic compound. At the beginning itself, we classified compounds as organic and inorganic. And students, you can remember any compound that does not have carbon in it, with the exception of oxides, carbonates and bicarbonates, were classified as inorganic compounds. And water is one of the main inorganic compounds, or we can actually say the main inorganic compound present in bodies of living organisms. So if we take the body weight, 2/3. If it is 100%, about 66 to 67%, that is 2/3 of the body weight of most organisms is by water. So the same thing, the main component of living organisms' body is water, and out of the body's weight, the 2/3 of it is the compound water. The rest of the body weight is only the other component. So that means water is present throughout the body of the living organism. And also water is an essential medium for the maintenance of living matter. To maintain the body of the living matter, that is for all the living processes to take place as well as the living organism to live in a certain place, it will need water. So, therefore, it acts as an essential medium. Life originated in water. This is another important fact. We talked about evolution. You can remember, students, from your previous grades. Evolution means the process in which the earth originated and developed, and all the living organisms originated and became very complex organisms. So, in that, the first step was the pre-cell, that is just one cell organism that evolved first. So, this evolved in water. It originated in water. So, water is a very, very important medium for living organisms. And the composition of water is very simple. If we write the molecular formula, you know it, H2O, 2 hydrogen atoms and 1 oxygen atom. So, it is a very simple composition. The chemical formula is also very simple and very easy to remember. As water is the most abundant inorganic compound found in living beings, it is important in many biological functions. So, since it is the main component and it is present everywhere on earth, it is very, very important for biological functions. Biological functions, the activities related to living organisms. For example, when you eat food, the digestion of food is a biological function. Growth of an organism is a biological function. Whereas reproduction, living, the habitat where the plant or animal lives in, all those, the growth, everything relates to biological functions. So, all these need water as the important component. Now, we will discuss the special properties of water that make it so useful for all the living organisms. So, specific properties of water and its contribution for the maintenance of life. So, specific properties and maintenance of life. So, we will look at each property and how it helps to maintain the life. The first property, it is a good solvent. As you know, in your household, if you want to dissolve something, say for cooking, making tea, making juice, you use water to dissolve a substance. So, the ability of a substance to dissolve another substance is called the solvent property. And out of all the solvents available, water is the best solvent, we can say, in terms of living organisms. So, here it is considered as a good solvent, very useful. Even for washing, many different purposes that are done in day-to-day life involve the solvent property of water. Similarly, this property helps living organisms in many ways. It provides a medium for biochemical reactions in the cells of organisms. Here we need to note down these words: medium, biochemical reaction, and in the cells of organisms. So, if you take a living organism, it can be unicellular, just one cell, or a multicellular organism with many cells. But all the cells have biochemical reactions taking place in them. You know what chemical reactions are? A change where there is bonding involved and the initial substance is converted to a new type of chemical substance. That change that is not reversible is called a chemical change. And when this change takes place in a living body, the cell of a living organism, then we call it as biochemical reaction. We discussed photosynthesis and cellular respiration when we talked about carbohydrates. So, you know what these biochemical reactions are. So, when these reactions take place in the cell, the reactants, for example, carbon dioxide and water in photosynthesis or glucose in cellular respiration, has to be present in a medium. So, they are usually dissolved in water. So, any substance that is going to undergo a chemical reaction is usually present in the water medium. Therefore, water provides a medium for biochemical reaction. The second one is the main constituent in extracellular fluids of organisms. Main constituent in the extracellular fluids of organisms. So, when you say extra, it is outside the cells. If you take the body of a living organism, it is made up of cells. It is the structural unit. Between the cells, there is space. In that space, there is a fluid. That is called the extracellular fluid. That helps to keep the cells moist, provides water to the cell, provides other nutrients to the cell, and acts as a certain medium. So, this extracellular fluid has water as its main constituent. Then the third one facilitates removal of excretory material and fecal material. So, when there are biochemical reactions taking place in the body, there are waste materials produced. When we say waste material, a chemical substance produced during the metabolic activity. Activity which is a chemical reaction that is taking place inside the cell is called the metabolic activity. So, the byproducts or the substances that are produced during a metabolic activity but are not needed for the body are considered as excretory material. For example, when we eat protein containing food, there is ammonia produced. So, this ammonia which is converted to urea or uric acid is not useful for the body. It can become harmful at times. So, therefore, this material has to be excreted from the body, and that is called an excretory material. This is excreted with urine. The nitrogenous waste materials are excreted with urine of organisms, and the main component of urine is water. So, because water can dissolve the nitrogenous waste materials, it is excreted easily from the body. That is one. Then we have the fecal matter of animals. As you know, students, animals consume food. They need to get their energy by eating or consuming food. So, this food gets digested within the digestive system. But there are some parts of the food that do not digest. So, these undigested food particles along with other chemical substances is known as fecal matter. This fecal matter is not an excretory material. There are two different things. Fecal matter is not an excretory product. You have to remember that. So, that is only undigested food, but that also has to be sent out of the body. So, for that, the removal of feces has to be carried out, and water is a main component that helps to remove feces from the living organism's body. So, that is possible because of the good solvent property of water. And last, important in respiration of aquatic organisms as oxygen is soluble in water. So there are gases that can dissolve in water and oxygen is one such gas that can dissolve in water. And as you know students, oxygen is important for living organisms because we breathe in oxygen and this oxygen is used in cellular respiration in order to produce energy for the function of the living body. So to do that, all the organisms need to breathe oxygen. As humans, we breathe air from the atmosphere. Like us, all the organisms that live on Earth's surface, that is the soil surface or what we call as the terrestrial environment. We breathe oxygen from the atmosphere. But there are animals and plants which are known as aquatic organisms because they live in water. Aqua means water. Aquatic organisms that live in water. These organisms need to inhale or breathe in oxygen from water. For example, if you take the fishes living in water, they breathe through their gills. They take in water, absorb the oxygen, and then water comes out. So, to this process to be successful, oxygen has to be dissolved in water. So, the respiration process of aquatic organisms is possible because oxygen is dissolved in water. So, that the good solvent property of water helps in many ways for the living organisms. We will go on to the second property. It is a coolant. Coolant means a cooling agent which helps to reduce the temperature of the bodies of living organisms. We will see how. Now this water has high specific heat capacity. When we say specific heat capacity, if we take 1 kilogram of water and if we want to increase its temperature, let's say from 30 degrees Celsius to 31 degrees Celsius. So there is an increase of 1 degree Celsius and it is 1 kilogram of water. We need to provide heat. So the amount of heat needed to increase temperature by 1 degree Celsius in 1 kilogram of mass is called the specific heat capacity. And the specific heat capacity of water, you will learn the value in other lessons later, is 4200 joules per kilogram per Kelvin. So this is a very high value. And because of this high value, water doesn't become hot very quickly. That is why the sea does not become hot during the daytime quickly like the sand or the soil. So that is why it helps to cool the organisms. So because water is inside the living body, it absorbs lot of heat, it needs lot of heat to become hot. It helps to maintain the body temperature of living organisms. So here you can see the body temperature does not fluctuate quickly with changes in the environment. You all know birds and mammals, that is humans, us and the other mammals, we are warm-blooded animals. Warm-blooded animals means the body temperature does not change with the change in environment temperature. Whether the environment is cold or hot, our temperature is maintained at a certain value. So to do this, water is very, very helpful. The high specific heat capacity of water makes it a good coolant. So because water is a good coolant, we are able to maintain the warm-bloodedness of our body. Now we'll go on to the next property. The next property, high cohesive and adhesive force. When we say cohesive force, it is the force between two particles of a compound. Here, when we take water, between water molecules, there is an attractive force which helps the water molecule to be a continuous column of water. And that force is called the cohesive force, force between the constituent particles. And the adhesive force is when water is flowing through a tube, whether it's a straw or within the plants or within the animals. When water is flowing, there should be a continuous column. For this to happen, there should be an attraction between water particles and the wall of the vessel. So, this particular attraction is called the adhesive force. So, the cohesive force as given here is the attraction between water molecules, and the adhesive force, water and another molecule attraction. Due to the high cohesive and adhesive force of water, it helps water to flow as a continuous column. Now we will see how this helps living organisms. Being the main constituent of blood, it helps transport nutrients, vitamins and hormones to relevant locations. So, as you know, these are three types of substances, nutrients and vitamins as well as hormones. All these have to be circulated with blood throughout the body, and blood flows through blood vessels. So, as we discussed before, the blood has water as its main component, and there should be the cohesive force between the water molecules and the adhesive force between the water molecules of blood and the wall of the blood vessels, so that it can flow easily as a continuous column of blood. So, this helps to transport all the main substances to the relevant locations. Similar to that, in plants, transports water to the upper parts of the plant due to high cohesive, that is water-water attractions, and adhesive water, another molecule attraction, force of water molecules. So, in plants, you know students, the roots absorb water from the soil. So, that is the ground level. From the ground level, water has to be transported to higher parts of the plant, especially the leaves where photosynthesis takes place. And if it is a very tall plant, the water has to be taken up with very high force, and that also against the gravity, the pull of the earth. So, for this, the high cohesive and adhesive forces of water helps in pulling the water up through the transport tissue. That is the xylem tissue. In a plant stem, there is a tissue called the xylem through which water is transported, and the adhesive and cohesive forces of water help water to flow continuously from the root to the top of the plant. So, that is this property. Then the next property is called differential expansion in freezing. Now, all substances have a density. You all know density is related to mass and volume. And water, that is the liquids, when you decrease the temperature at a certain temperature, they freeze and become solids. So, water freezes and becomes ice. And when it does, the density of water is high and the density of ice is less. So, when water turns to ice, the density of ice becomes less. Therefore, ice floats on water. Water will be below and ice will be floating on water. This is very, very useful for aquatic organisms living in cold countries. When ice is formed, they come to the top layers of water, keeping water as it is in the bottom. This provides living environment for aquatic organisms. Unlike our country, in certain other countries, for a certain period of time, there is snow. So, the water turns into ice. When that happens, in the water body, only the surface water will become ice, and because of the less density, it will float on ice, but below there will be liquid water. And all the living organisms which were present in that water body, like the aquatic plants and animals, survive that particular cold season, being inside water. So, that helps in the existence of aquatic organisms. And this property of water, where the density of ice is less than the density of water, is called differential expansion in freezing. The density of ice becomes less because it expands more than water. Okay, so these are different properties of water that helps it to be an important component in the bodies of living organisms and to maintain the life of living organisms. So, I hope students that you got a very good understanding of the different properties of water and how they are useful for the maintenance of life. So, that is one of the main inorganic components. The next main inorganic component is minerals. Now, we will discuss about minerals. Minerals. Minerals are important as a nutrient constituent to maintain the life process of organisms. So, here they are nutrient constituent. So, they have a nutritional value. When we say nutritional value, the presence of that particular substance is important for the proper function of the living body. That is what we mean by nutritional value. So, here to maintain the life process of organisms, they are important. They are absorbed as trace or macro elements. So, based on the amount of element absorbed, we saw that there are many elements. These come under inorganic elements, and some are absorbed in high amount, some are absorbed in less amount. So, based on that, we group them as trace or macro elements. And the elements needed in higher amounts are known as macro elements, and the elements needed in small amounts are known as trace elements. So, if the living body needs a lot of that element, it is known as macro. Macro means large, macro element. And if only a very small amount, say in milligrams or micrograms or very tiny amount is needed, then we call it as a trace element. 7% of the body weight is by minerals. Now, we were discussing the composition of the body of living organisms. In that, 7% of the body weight is composed of minerals. Only a small amount, but it is very important. 3/4 of the above amount, the 3/4 of this 7% is composed of calcium and phosphorus. So, these are the two main minerals that are there in the living body, calcium and phosphorus. Other than that, potassium is there, iron is there, magnesium, copper and iodine are there in a living body. Potassium, iron, magnesium, copper and iodine are present in the living body. When the elements are not present in correct amounts, so we need them in a small amount, and under that also some are macro, some are trace element which are needed in very small quantity. But still, if they are not present in correct amounts, both plants and animals show deficiency symptoms. So even that small amount is not present in the body, then there will be an imbalance in the body. That will lead to certain deficiency symptoms. So here we need to understand these two words. Deficiency means it's not there in the required amount. The amount does not matter. It can be a high amount or a small amount, but if the required amount is not there, then we call it deficient. And symptoms are the features that you can see due to this deficiency. Say if the skin becomes pale in color, then that can become an deficiency symptom. You see the difference in the color, dryness in the skin, that is a symptom. So what you see from the external body, that is a symptom caused by the deficiency of the mineral. So now we have discussed the importance of minerals, the trace and macro elements, and the different elements that are present in living organisms. Now we will see how these minerals are important for the existence of both plants and man. First, we will see the functions of minerals in human body and the deficiency symptoms of these minerals. Under that, we look at the first element, that is potassium. So when we talk about potassium, this controls the ionic balance of the fluid in the cell. If we take the human body, it is made up of cells and there is fluid inside the cell and as extracellular fluid. And the main component of these fluids is water. And in water, there are minerals dissolved. So these minerals are the ions present in the body. And there should be a balance between all these types of ions. And potassium helps to maintain and control the ionic balance of the fluid in the cell. The second one, it is important for the activity of heart and muscles. You know that when the heart pumps blood only, the blood will be circulated throughout the body. So the proper functioning of the heart, the beating of the heart is important for the circulation of blood. So the activity of heart and the muscles, all the muscles are controlled or helped by the presence of potassium in human body. Then transmission of nerve impulse. So as I am talking to you, you can hear the sound. This sound is a stimulus. It is received by your ear and the information is carried to your nervous system and the brain, the part of the nervous system, the brain. There you get the information. So the sound, what you hear from your ears has to go to the brain. So this transmission is done by the nervous system of our body. And the way this information is transmitted from one place to the other, the sensory organ to the nervous system, is called transmission of nerve impulse. The information is transferred as impulse. So transmission of nerve impulse is done by potassium mineral. So potassium is important for the transmission of nerve impulse. Therefore, if this potassium element is not enough in the body, there will be these deficiency symptoms. The muscles of the body can become weak. So, weakening of muscles and also psychological disorder. Psychology means the mood, the mental state of a person can change due to the deficiency of potassium as mineral. Now, we will look at the next mineral, sodium. So, sodium is necessary to activate enzymes. Now, you can remember, students, what is an enzyme? Yes, it is a special protein that is produced within the body in order to do what? To increase the rate of biochemical reactions. So, special type of proteins produced in the body to increase the rate of biochemical reactions are enzymes. And this activity of the enzyme is done by sodium. Sodium activates enzymes. So, then only they can function properly. And sodium is a constituent of digestive juice. When you eat food, the food has to get digested, and this happens inside the digestive system where the food is mixed with a liquid substance, and that is called the digestive juice. So, sodium is a constituent of the digestive juice. Then we have the next function to maintain constant osmotic pressure in cells. Now, the human body is made up of cells. Inside the cell, there is certain amount of water and ions. We saw that before. There is water and iron. And there is transfer of water and iron into and from the cell. So, this is controlled by a factor called the osmotic pressure. A certain pressure within the cell due to the water and ions present. So, this is maintained by the presence of sodium ion. And finally, transmission of nerve impulses is done by sodium. So, in a human body, there is nerve impulses transmitted throughout the nervous system, and that is done by two minerals, potassium and sodium. Both these are needed for the transmission of nerve impulse. These are the important functions of sodium. So, if sodium is not present in enough amount in the human body, these deficiency symptoms can be observed. The first one is the respiratory disorder. It can be difficult for you to breathe in and out, inhale and exhale properly. So, something wrong with the respiratory system. That is called a respiratory disorder. Cramps. When you all do activities, lot of physical activities, you get muscle cramps, say during sports activities or some other heavy physical workout, you can get cramps. The reason is there is not enough sodium in your body. When you sweat a lot, you lose a lot of sodium with sweat. Then also there can be a deficiency in sodium. So, to get rid of the cramp, what do? We take a little bit of salt. That is to provide sodium. So, cramps is another deficiency symptom of sodium. Another symptom is nausea. Nausea means feeling vomitish, having a headache or turning off the head, feeling uncomfortable, an uncomfortable feeling in your stomach. All these are part of the nausea symptom. So, that is also a symptom of deficiency of sodium. And finally, it can even lead to diarrhea. So all these are the symptoms of sodium deficiency. And if you think you have these symptoms, then you might have to consider taking more sodium with your diet. So these are the first two minerals. Now we will go on to the next mineral that is important. The third one is magnesium. Magnesium is Mg. It is a constituent of bones and teeth. Bones and teeth are the strongest components of a human body, and these two contain magnesium as a constituent element. To control nerve activity. So here it is only to control nerve activity in skeletal muscles. There are different types of muscles in the human body. The muscle that is attached to the skeleton. Skeleton means the bones of your body. This is important for the movement of your limbs. Say the hands or the legs when you walk, when you write, when you move your hands, when you are talking. All these are done with the help of skeletal muscles. So the muscles attached to the skeletal system of the body. To control the nerve activity of the skeletal muscles, there should be magnesium in the body. Otherwise, you will not be able to move your limbs properly. Then the next one is help in metabolic activities. When we say metabolic activities, these are the chemical activities that take place in a living organism. Making of substances or breaking down of substances. All these together are known as metabolic activities. So these are the functions of magnesium ion, and if this is deficient, we will see these two deficiency symptoms. High heartbeat and nerve irritability. High heartbeat and nerve irritability. Because this magnesium is needed to control the nerve activity, there can be nerve irritability. Nerve irritability, you feel a sensation, maybe the tip of your fingers or your hand or the sole of your leg. You can feel a burning sensation or a sensation irritability. All that is due to the deficiency of magnesium. That is possible. And also the heartbeat can increase more than the normal rate. So these are two symptoms caused by the deficiency of magnesium. Now we will look at calcium. Calcium again is another mineral needed for the proper functioning of human body. It is required for the growth of bones and teeth. As you can see here, magnesium is a constituent of bones and teeth. Calcium is needed for the growth of bones and teeth. So if you want to have strong teeth, strong bones, for the development, you need to have enough calcium in your body. Then it is important for blood clotting. When you have a wound in your body, a small cut in your hand, there will be blood flowing out. If we don't stop the blood flow, then there can be excessive blood loss. This is prevented by the body by the process of blood clotting. So for the blood clotting to happen properly, there should be enough calcium in the body. So the calcium ion prevents excessive blood loss due to injuries, helps to prevent blood clotting. And proper function of nerves is also controlled by calcium, or calcium is necessary for the proper function of nerves. It is needed for milk production and also for the absorption of vitamin B. So the nerves transmit impulses, and they are important for the coordination process and receiving senses and getting information from that. So all that is done by the nerves. And for the function of the nerve, we need calcium. Then milk production. A newborn baby drinks milk from the mother, and the milk has to be produced within the mother's body. For this to happen properly, there should be enough calcium in the mother's body. And the final one is absorption of vitamin B. So even though you eat food containing vitamin B, you need enough vitamin B for the function of the body, and you do eat a lot of vitamin B. But if you don't have enough calcium in your body, the vitamin B will not be absorbed by the body. So then it will not be useful for the body. So therefore, for the absorption of vitamin B, we need to have enough calcium in the body. Since these are the functions of calcium, if calcium is deficient, we will get these types of deficiency symptoms. The first one is weakening of bones and teeth. Since calcium is needed for the growth of bones and teeth, if that is not enough in the human body, the bones and teeth will become fragile. They will become weak. So then they can break easily. The second symptom is growth disorders. The calcium ion is needed for the proper function of nerves, milk production. So if it's not enough in the body, it can lead to problems in the growth. So due to that, a child may not grow properly. So that is what we call as growth disorders. And finally, osteoporosis. Osteoporosis is a common disease nowadays. Osteo means bones, porosis means pores or holes. So if we don't have enough calcium in our body, normally also the human bones have small pores, small holes in it. If there is not enough calcium, these pores become large. So the bone will have lot of large holes in it. Therefore, the bone will be weak. And even if you fall down, light fall can lead to a crack in the bone. That is the effect of osteoporosis, which is caused by deficiency of calcium. So as we have seen, magnesium and calcium are two minerals that are also needed for the proper function of a human body. Now we will look at the other elements. Phosphorus. Phosphorus, P. This is needed for the growth of bones and teeth. Phosphorus is also important for the growth of bones and teeth. And it is a constituent of nucleic acid. Remember students, we discussed this before. Out of the 4 biomolecules, the nucleic acids only have phosphorus as a constituent element. For the carbohydrate and fat metabolism. So as I said before, what is metabolism or metabolic activity? Yes, it is the making or breaking down of chemical substances within the living body, or the chemical changes or reactions that take place inside the cell of a living body. So for these metabolic activities, especially the metabolism of carbohydrate and fat, there should be enough phosphorus in the human body. The third one, instant release of energy in muscles and nerves. So, instant release of energy in muscles and nerves. For you to function quickly or actively, if you are a sports person, when you are doing some sport activity, or if you are doing your normal day-to-day activity where you need to move a lot and fast. Then your muscles and the nerves have to coordinate together and move properly. For this to happen, there should be enough energy within your muscles. So even though you eat properly, you have a carbohydrate storage in your body. If you don't have enough phosphorus, then the energy will not be released immediately whenever necessary. So that can lead to certain deficiency symptoms. So if we look at the deficiency symptom, the main deficiency symptom caused by phosphorus deficiency is the weakening of bones and become fragile. The bones become weak, lighter. Fragile means they can break easily. So that is the symptom. Then we will look at the importance of iron. Iron again is another mineral, Fe, and it is very, very important for the synthesis of hemoglobin. Hemoglobin, as you know, is the main component of red blood cells. So, the hemoglobin is the substance that helps to transport oxygen throughout the body. So, this iron is important for the synthesis of hemoglobin. And if that is not there, that is iron is not enough in the human body, there will be not enough hemoglobin present in blood. This condition is known as anemia. And due to this anemia, we have certain symptoms, that is the sleepiness, hypoactive nature, and weakness in psychological development. So, if you don't have enough iron in the body, there won't be enough hemoglobin in the blood. So, the amount of oxygen transported throughout the body will not be sufficient for the proper functioning of the human body. Therefore, you will feel sleepy all the time. You won't feel like doing work actively. You have a lethargic nature. You feel tired and you don't feel like moving. You don't feel like studying or doing sports activities. That is what we call as hypoactive nature. And all these can lead to weakness in psychological development. You are not very active in your mind as well. So, the psychological development can become weak. Another function of iron is the storage of oxygen in muscles. So, as it is important to produce hemoglobin, iron is important to store oxygen in muscles. Inside the muscles, there is glycogen, there is oxygen, and whenever necessary, glycogen will be converted to glucose and that will react with oxygen during cellular respiration and that will produce energy for the function of the body. For this, we need to have enough iron in our body. And the last importance is constituent of enzymes. We have already talked about enzymes. They are the biological catalyst. They are needed to increase the rate of biochemical reactions. And for the function and to make the enzymes, because iron is a constituent, it is part of the enzyme. We need to have enough iron in our body. The next one is iodine. Iodine is important for the synthesis of thyroxine hormone. We already talked about hormones. There are lipid hormones and protein hormones in the body. And these hormones are chemical substances needed for the coordination and homeostasis. Coordination is the proper regulation and the co-acting of all the parts of our body. Say if we want to write something or if we want to say a word, the hands or the muscles of the mouth have to function properly. And also the environment inside the body has to be maintained in a certain manner. All these processes together is called coordination. And maintaining the proper environment inside the body, which is called the internal environment of the body, is called homeostasis. For these two, the thyroxine hormone is important, as well as the growth and the proper function of the human body. The metabolic activities of the body are also controlled by thyroxine hormone. So, for the synthesis of thyroxine hormone, we need to have iodine. That is why most of the salt that we get in the shops contain iodine. So, for most people, we need to take enough iodine so that enough thyroxine hormone is synthesized in the body and it controls the functions of the body. But if you are a person who has too much of thyroxine, then of course the person has to reduce the intake of iodine. So normally a healthy person needs a certain amount of iodine for the proper function of the body. And if iodine is not enough, that is deficient in our body, we will get these deficiency symptoms. It will affect the development of intelligence. The normal intelligence of a person can be affected due to the deficiency of iodine. Then there will be a lethargic attitude towards studies. So students, this is very important to y'all. Y'all are at the age where you have to study more. Your main object is to do your studies well. So if you don't have enough iodine, you will have lethargic attitude towards studies. So you need to prevent that by having enough iodine included in your food. And finally, it limits body height. So because thyroxine is important for the growth of the body, if you don't have enough iodine, there won't be enough thyroxine. Therefore, the body height will be limited. So these are the deficiency symptoms that you will see in a person who is deficient in iodine. So that brings us to the end of the importance of different types of minerals for humans, that is us. Next, we will see how these minerals are important for the proper functioning of a plant. The functions of minerals in plants and deficiency symptoms of them. So, the first mineral is nitrogen. It is as a constituent of amino acid, proteins, nucleic acid and chlorophyll. So, we have already discussed about amino acids. Those are the constituents of proteins and nucleic acid, which is important in the field of genetics, and also the chlorophyll in plants. So, as you already know students, proteins and nucleic acids are biomolecules and amino acids are the basic structural units of proteins. So, nitrogen is a main constituent of all these components. Then if we look at chlorophyll, it is the green color pigment that is present in green plants. And it is important for the process of photosynthesis. Actually, photosynthesis takes place in the chlorophyll pigment in green leaves. So, if nitrogen is not enough in a plant, there will be these deficiency symptoms. The retardation of growth of the plant. So, that means the plant will not grow as expected. There will be not enough height or it will not produce enough fruits. So, that is what we call as retardation in growth of the plant. Then there is an important symptom which is called chlorosis. This comes from the word chlorophyll. So, if there is enough chlorophyll in the plant will have a proper green color. But if there is not enough chlorophyll, the color of the plant will change. It will become a pale color, or there can be yellow or brown patches. So, this particular condition where there is not enough chlorophyll in the plants is called chlorosis. Chlorosis in mature leaves is a deficiency symptom of nitrogen deficiency. So, chlorosis in mature leaves. Now, we will look at the next mineral, that is phosphorus. So, phosphorus again is as a constituent of nucleic acid and ADP, adenosine triphosphate. So, nucleic acid is a biomolecule that you already know about. Then there is something called ATP, adenosine triphosphate. We have already discussed the process of cellular respiration when we discussed carbohydrates. So, this is the process where glucose molecules are broken down in order to produce energy for the function of the cell. So, in that the energy is produced and contained within molecules called ATP, adenosine triphosphate. So, the phosphorus element is a constituent of ATP. Then, due to the deficiency of phosphorus, we can see retarded growth of roots. The root will not grow properly. Therefore, the plant will be unable to absorb enough water and nutrients for its function. And red and purple color patches on leaves will be visible. There will be patches. The normal green color leaf will have red and purple color patches on it. So, that is the deficiency symptom caused by phosphorus. Then again we have potassium. Potassium is another mineral important for the plants. It is important for protein synthesis. Inside the plant cells, there is protein synthesis taking place. That is the production of protein within the cell. For this to take place, there should be enough potassium in the plant body. Then the opening and closing of stomata. If you take a plant leaf, the surface of the leaf has small openings called the stomata. This is the opening through which air is exchanged. That is, the carbon dioxide that is needed for the plant for photosynthesis goes into the leaf through the stomata. The same way, the byproduct of photosynthesis, oxygen, is given out of the plant through the stomata. So, stomata is the pore present in leaves or throughout the plant. There are stomata in the stem as well. This helps, or this is important for the gas exchange process. So, the opening and closing of stomata will not take place properly if the plant has potassium deficiency. So, due to the deficiency, we can see these two symptoms. Chlorosis, as we discussed before, in leaves. So, the reduction in green color, chlorophyll pigment in the leaves will be a result of potassium deficiency. And also formation of yellow or brown patches in the leaves can be observed. So, these are the deficiency symptoms due to potassium. Next is iron. Fe. This is again important for synthesis of chlorophyll. So as you already know, chlorophyll is an important pigment which gives green color to the plants and it is necessary for photosynthesis. So if the plant body does not have enough iron, then there won't be enough chlorophyll. Then the photosynthesis process can be affected due to this deficiency. And synthesis of respiratory enzymes. Students, you all know that green plants have photosynthesis process during the availability of sunlight. That is, during the daytime they do photosynthesis, but night time they do respiration. So for this respiration, there are enzymes needed inside the plant body. So for these enzymes to be synthesized or produced within the plant body, the plant has to have enough iron. And if iron is deficient, then there will be chlorosis in tender leaves. So if you can remember, we discussed in the previous slides also, chlorosis is the deficiency symptom where chlorophyll pigment is not there in the plant or it is not enough in the plant. But this chlorosis can be in the mature leaves, tender leaves or any leaf. So here deficiency of iron, chlorosis is seen in tender leaves only. Okay, we will look at the next element that is necessary for plants. It is calcium. So the first one, it is important because it is a component of the cell wall. So as you know students, in a plant cell, there is the cell membrane. Outside the cell membrane, there is a cell wall. What is the main component present in a cell wall? Yes, it is a carbohydrate and it is cellulose. Cellulose is present in the wall of plant cells. In addition to that, there is calcium present. The second importance is to maintain the structure and function of plasma membrane. That is the second importance, to maintain the structure and functions of plasma membrane. If we take a plant cell, this will be the shape. Outside is the cell wall and inside we have the cell membrane. Within the cell membrane, all the organelles of the cells are present in the plasma. So this is the plasma and this component is called the plasma membrane. So for the structure and functions of the plasma membrane to be maintained properly, we need to have enough calcium in the plant body. The next one, for the activity of enzymes. So the calcium ion is also needed for the activity of enzymes. You already know what enzymes are. They are biological catalysts, and for the activity we need to have enough calcium in the plant body. So if calcium is not enough in a plant body, the following deficiency symptom, that is the dying of tissues at the tips of the leaves. This will be observed in a plant body. If you look at the leaf, the tip, when you have the certain shape of a leaf like this part is the tip of the leaf, that will die. So you can see it as a brown colored tip which is curled and not very fresh. So that is the dying of tissues at the tips of the leaf. The next element, zinc. This is necessary for the activity of most enzymes. So as you already know, the biological catalysts are enzymes. For their activity, we need different types of minerals and one of those is zinc. And also it's necessary for the synthesis of chlorophyll. Like other minerals, zinc is also necessary for the synthesis of chlorophyll. So, if this is deficient in the plant, there will be dead cells and tissues throughout the plant. So, if it is a normal plant, a healthy plant, you will see that the stem, the leaves, everything will be fresh. But if the plant is deficient in zinc, then you can see there will be dead cells. You will see brown color, dark brown color patches where it's very dry because of the dead cells. And also there will be extra thickness in the leaves. Normally the plant leaves are very soft and nice. Here there will be extra thickness. As you look at the leaf itself, you will be able to identify that there is a difference in the texture. It's too thick. So that is due to deficiency of zinc. Then we have sulfur. Sulfur is S and it is a constituent of amino acids and therefore proteins. You all know the constituent elements of protein: carbon, hydrogen, oxygen, nitrogen, and there is sulfur in some of the amino acids. So, any sulfur containing amino acid, if it is present in protein, that protein also will have sulfur in it. So, it is a constituent of both amino acids and proteins. And if sulfur is not enough in the plant, the deficiency symptom will be chlorosis in veins and areas between veins. So, if we look at a plant leaf, it has veins like this. Different types of venation is there. So, here chlorosis in veins, this part, and between veins. So, those are the veins of the leaves. So, as you can see, there is chlorosis caused due to different deficiencies, and if it is iron, chlorosis in tender leaves. If it is sulfur, chlorosis in veins and areas between veins. So, students, you have to remember different mineral deficiencies cause chlorosis, but you have to remember in which part of the plant leaf the chlorosis is observed. Okay, so with that we will go on to the next slide where you can see the deficiency symptoms. Deficiency symptoms in plants. So, this is the first one where you see the nitrogen deficiency symptom. You can see it is chlorosis in mature leaves. Mature leaves is the grown up. The new leaves are known as the tender leaves, and with time they become the mature leaves. So, chlorosis is this part becoming, you can see these areas becoming yellow in color. The normal green color is not there. So, that is what we call as chlorosis because of nitrogen deficiency. Then, zinc deficiency causes extra thickness in the leaf. If you look at this leaf, it looks different from a normal leaf. This looks more thick. So, that is due to zinc deficiency. Then, phosphorus deficiency causes red. You can see all these dots. Those are the red and purple patches in the normal leaves. Remember, students, there are some plants, for example, croton, which has a normal mosaic leaf. That is the nature of that plant, croton. But if it is a normal leaf which has red and purple patches on it, then that means it's a phosphorus deficiency symptom. Then the next picture shows the potassium deficiency symptom where you have yellow and brown. Here you can see the outer edge of the leaf is dry and brown in color, and inside there is yellow color area. So, yellow or brown patches in leaves is a deficiency symptom of potassium deficiency. The last picture shows you the calcium deficiency symptoms. Here you can see the dying of the tips of the leaves, dying of tissues at tips of the leaves. This part is dry and dead. So, that is caused by calcium deficiency. Students, if you have a look at these pictures, you can get an idea as to the deficiency symptoms caused by deficiency of minerals in plants. So, when you go to the environment, say you have a garden or you go to a garden where there are plants, and when you look at the plant, if you see any of these symptoms, then you can identify the cause of it. Why the leaf is like this? Is it chlorosis, or is it dying of tissues at the tips, or is it the brown and yellow patches, and what has caused this symptom? Okay, so with that we come to the end of the discussion regarding minerals and their importance to humans and plants and the deficiency symptoms caused to these plants and people if it is not present in the required amount. So, with that, we will go on to the next type of compound that is important for living organisms. This is an organic compound and it is vitamins.